Our next speaker is also going to join us virtually, not quite from so far away in North Dakota, but a Manitoba farm boy. Dr. Barney Geddes was in a farm in central South Manitoba. He actually was our student at Crop Diagnostic School in 2007, along with Colin Penner. So, but after that, he completed his PhD right here at the University of Manitoba, doing work with Rhizobium legume interactions. Then continued on, did work in England, University of Oxford, again, looking at the Holy Grail, nitrogen fixation by cereal crops, and then follow-up work in McMaster University. But he's had his own lab and activity underway in North Dakota since 2020. Now, the reason Barney can't be with us today is he doesn't quite have his green card in order, we're told. We'd expected to have him here. And I could have fixed that because some people know that I had a stowaway cat that went across the border in November, and it now is in Grand Forks. So I could have solved that for you, Barney. But anyways, we'll let you take it away with your topic here today, biological potential to meet crop nutritional needs.
Great. Yeah, you can hear me okay?
Loud and clear.
Good. Yeah. Well, thank you for that kind introduction, John. Yeah, as John mentioned, I'm a Manitoba boy, but ironically, I'm stuck in the U.S. A little longer than I expected because of a problem in the visa application process to kind of settle down here. Nevertheless, I'm really pleased to have been invited to present at this conference. It's a little bit different than conferences I've presented at in the past. I was really stuck into a lot of basic research in my past. But when I started my group at NDSU, coming from a farming background, one of the things I was really keen to do is make sure we did some work that can get some information and outputs to farmers right away and have this kind of nice closer connection to the farm with our research. So my group down there, we call the Geddes lab. I've got the cool one written here because I understand my brother Charles presented here last year. So I just wanted to make sure everybody's aware. This one at NDSU is the cool Geddes lab. We study microbiology and especially microbiology to deliver nutrients to crops and improve crop growth. So I'm going to talk about biological potential to meet crop nutrient demands. And I'm really just going to focus on one nutrient, which is nitrogen, the one that's the most important in my point of view. So let's jump into it. I like to start with this infographic I discovered earlier this year with this giant green box you can see that says synthetic fertilizer. So this infographic is actually life-saving inventions, the most life-saving inventions science has brought to us. And how impactful those have been. And I like to show these to the healthcare researchers because here fertilizer has been a more impactful invention in terms of saving lives than all of medical science put together. So I think that's sort of entertaining. Some people may find that to seem a little biased. And as you all know, it all just depends how you put the numbers together. In this case, the reason that that's so outstanding is because in truth, half of the world is actually fed by synthetic fertilizers. So they're so crucial to yield in agriculture to allow our farmers to feed the world that if we lost those tomorrow, half of the world would go hungry. And so that's those deaths prevented that they were showing in that previous graph and why nitrogen fertilizer might be the most life-saving invention of all time. Now, of course, we weren't always this dependent on nitrogen fertilizers. This is an issue that grew over time. The more we used them, the more population expanded, and now we're quite dependent on them. Nevertheless, they're a crucial portion of our farming system now, and they're feeding the world. Now, the problem is that probably any of the farmers here today will know that they're getting really, really expensive. They probably, in many cases, were already the most expensive input for a lot of farmers in the spring. And this graph that I pulled last year in March, at that point they'd actually tripled in price from a couple of years before, which is just astronomical. And you can see how that would really quickly cut into the bottom line for farmers. So cost of nitrogen fertilizers is starting to become a big issue. The war in Ukraine's put even more pressure on that, like it has on other things. And in addition to that, Canadian politics are starting to get involved as well, right? So I'm sure many people are aware of this news story that came out, or this 30% reduction in fertilizer emissions target by 2030 that the Canadian government was proposing. So I don't think there's any, hard stuff coming out about that yet. And there was certainly a big backlash by farmers because they know they need these fertilizers to compete on a global market. But I think we have to acknowledge that their use may be restricted in the future. So we've got this duality of expensive fertilizers, but also, we may not always be able to rely on having those to maximize our yields going forward. So really the pillar of my research program, and also I view the pillar of sustainable agriculture, are legume crops. So these are, things like peas and beans and chickpeas and lentils and even soybeans. And the reason for that is because they actually don't rely on nitrogen fertilizer, at least not nearly as much as other crops, because they're able to engage in this really miraculous symbiotic relationship where they can form new organs on their roots, these sort of bright pink root nodules that you can see in the picture I've shown here from a pea plant I pulled up from our farm a few years ago. Inside of those nodules, bacteria called Rhizobia live in there in huge numbers, millions and millions of these Rhizobia in each nodule. Inside the plant cells actually can actually take dinitrogen, which is normally inert. It's the same dinitrogen we fix in factories to make synthetic nitrogen fertilizer, and transform that into usable forms for the plant. And what's really important here is that this is not producing any, it's not requiring inputs the way that chemical nitrogen fertilizer production requires. Chemical nitrogen fertilizer production uses like 2% of the natural gas in the world, produces a lot of CO2. But also the nitrogen is used directly by the plant for biomass. So we don't have any issues with unused fertilizers running off into lakes and streams or turning into greenhouse gases or anything else like that. So this is really a sustainable source of nitrogen for our crops. Now, we've been aware of this for a long time. I mean, there's some evidence even the ancient Romans knew something about how rotating with legume crops was good for their soils. In modern agriculture, we can actually apply these bacteria as inoculants to our legumes. And through doing that, we can grow seed crops and get basically sustainable proteins for consumption, but we can also grow alfalfa or forages for food for livestock. And we also can incorporate nitrogen into our agricultural systems for downstream crops that might not be able to do this symbiosis. So it's a really important and fundamental relationship to sustainable farming. So in this talk, I'm going to talk mostly about this Rhizobium legume symbiosis because that's what a lot of our research is on. But I'm also, John also asked me to talk about nitrogen fixation in cereal crops, something that I've studied in the past that we're continuing to start to study here at NDSU, but don't have anything quite ready to present. But particularly about the different products that are starting to become available for farmers. So I'll try to dive a little bit into that and what those are doing and just give you my perspectives on those as well at the end of the talk. But first, I want to talk about our work towards maximizing Rhizobia in legumes. The agronomic outputs from our program really focus on sort of two major questions. One is, can we actually guide farmers and give them more information about when should they actually be inoculating their fields and when can they be expecting to see benefits from inoculation? And the second is, can we improve on the technology that's out there? How can we actually maximize the benefits we get from inoculation, perhaps through developing new technologies? So before I dive into the research, just a quick primer on rhizobial inoculants. So different legume crops are nodulated by different rhizobia. So rhizobia is a broad term that encompasses a whole bunch of different microbial species. And they tend to be quite specific in their partnerships with specific legume crops. So they're not cross-compatible. Soybeans associate with Rhizobia of the genus Bradyrhizobium. Chickpeas associate with Rhizobia of the genus Mesorhizobium, et cetera, et cetera. So they're not cross-compatible. Each crop, really, the relationship is sort of unique to that. And I'm starting to feel that actually each one needs to be studied independently for these types of questions I described previously, because there's different dynamics occurring with these different pairs of hosts and symbionts. Now, when farmers go to buy products to apply to their seeds at planting, there tends to be different types of products available. Not all these types are available for all the different rhizobia. Some are more sensitive than others to the different formulations, but Generally, you can get your Rhizobia as a dried up granular inoculant, mixed into kind of more of a moist peat inoculant, or as a liquid inoculant that you actually got to pour right on the seeds almost right before you plant them, because they don't last very long once you put it on the seed as a liquid. So we have these different options available. And getting into the first question then, one of the things that really has been on my mind a lot is, should farmers inoculate? So maybe some of the agronomists in the audience have been faced with that question from farmers: should I be inoculating my crops? And I think different people have different responses for different reasons, and usually justified in one way or another. Some people might just say, yeah, you should always inoculate. Inoculants are wonderful. You get this great benefit. Some would say, I don't really tend to see benefits from inoculating. You probably shouldn't. Or others might just say that it's cheap insurance. So inoculants, compared to fertilizers, are really, really quite cheap. And maybe we should just be putting it on just because the consequences of not inoculating and expecting those Rhizobia to be there could be quite dire in terms of yield. So thinking about these different types of advice we can give in the context then of what we can know about a farm, and could we be more focused and a little bit more grounded or more justified in these types of responses for farmers? So what affects the success of inoculant? Well, one of those things is inoculation history. So just as an example, studies at the Carrington Research Extension Center that's in the Red River Valley here in North Dakota suggested that, at least for soybeans, you don't really need to inoculate for at least 5 years after you plant them for the first time. And so this is advice that's in our field production guide in North Dakota that says, as long as you've inoculated the first time you plant soybeans there, even up to 5 years later, you don't really need to inoculate again. You're not going to get a yield boost from inoculating. So the history of inoculation in the field is important. Now, remember, they're not cross-compatible, so it's crop to crop. And then there's a whole bunch of environmental variables that are going to be at play here in terms of the success of inoculants. So events like drought or flooding are harsh for Rhizobia. They can deplete Rhizobia populations. Soil conditions like acidic pH or salinity are also challenging for establishing symbiosis between the host and the symbiont. We've found that iron deficiency chlorosis in soybeans also seems to disrupt the mutualism in our work here. And finally, one that's not really maybe tend to be seen as a negative thing is high residual nitrogen. So this is something that's really important to keep in mind when you're planning what fields to be growing legumes in. If you have a high amount of residual nitrogen there, the plants tend to be quite good at detecting that they have this easily available free nitrogen in the soil, and they kind of get a bit lazy and tend to not form root nodules and utilize the Rhizobia like we might want them to. So all these things are important to take into account when we're thinking about inoculation. Now, in my group, what we're thinking about is precision agriculture and really building on this precision agriculture revolution and what I view as to be the new wave of that coming forward, which is incorporating soil biology information into precision agriculture. So what we would like to see is not just getting chemical analyses of farmers' soils and using those to guide decisions, but also to get biological analyses. Now, someday we might have some reliable soil health indices and other things that are much more complex to get to using microbiome information. But in the nearer term, we're focused on actionable information that we can give. And for us, that is how many Rhizobia for the target legume are present in your field soil. And hopefully we can use that to then guide the answer to that previous question of, should I inoculate it or not? Am I wasting my money because I won't see any response? Or should I definitely be looking at getting some inoculant on there to make sure that I'm getting this symbiosis occurring in my fields? We've been doing that in soybeans, peas and lentils, and chickpeas. And our approaches are relatively adaptable to others. And the way we've done that is actually building on work I did a little bit of at University of Manitoba, even before my PhD as an undergraduate researcher. Now we've kind of jumped back in and really pushed this technology to develop an assay where we can quantify the number of Rhizobia in a given field sample. The idea being that farmers can send the same soil in for a soil test chemically, and we can, for only a small additional cost, provide them information about how many Rhizobia are there present in their fields. So this is an assay that uses quantitative PCR. It basically quantifies the amount of DNA, specifically of the symbiotic genes of the target Rhizobia that are present in the soil. So it's really accurate. It deals specifically with the symbionts. And we can get to this information, and we've pushed the sensitivity pretty well. We're getting down to levels of about 100 Rhizobia per gram, which is really very few. We've also then been trying to figure out, well, where is the break point? Where's the line where we expect farmers to see benefits versus expect them not to see benefits to inoculation, so that we can be more accurate in our recommendations. We've done some work in the greenhouse looking at different levels of Rhizobia and where we stop seeing responses. For soybean, in our greenhouse experiments, it's above about 1,000 Rhizobia per gram that we stop seeing improvements from adding Rhizobia. So that's a little bit of calibration there, but we're moving now towards field trials to try to get a more accurate sense of what are the breakpoints where we can expect to see responses from inoculation. This is just one nice example of what this type of technology can do for us, for agronomists. So when we took and started to sample fields across North Dakota, and we went from this Red River Valley in eastern North Dakota down to over to our very dry soils in western North Dakota, we could see that this advice from Carrington saying that you don't really need to inoculate your soybeans for 5 years is not holding up. The Rhizobium populations are really, really low in western North Dakota. It seems to be associated with soil moisture, because when we check irrigated fields, they actually have plenty of Rhizobia there. And so this is kind of just an example of how these types of tools can accelerate our agronomy and just kind of get a clearer understanding of when inoculants should be applied or not. So to try to dive a little bit deeper in that, I mentioned we've started to do some field trials. So I'm going to tell you about a couple of trials we did last year in association with research extension centers here. And especially with Audrey Khalil and Mike Ostley and John Rickardson, who are scientists out at different research extension centers across the state. We asked the question of if there is residual Rhizobia present. And for peas and lentils and chickpeas, there really tends to be, especially for peas and lentils in almost any field. If there is Rhizobia there, do we see a benefit from applying inoculant? We had no inoculation treatments and liquid inoculant treatments and granular inoculant treatments. And we measured all sorts of parameters of these legumes, especially their nodulation and the yield, to try to answer this question. So just to show you, this is, we actually in North Dakota probably grow, we're leaders in all sorts of different legume production in the US. So I really love, it's a perfect place to have a research program that's interested in legumes. These sites were chosen to kind of span the different environments we might see across the state in terms of pulse growing regions. So they do a nice job being in very different conditions at the Carrington, Hettinger, and Williston Research Extension Centers. And this is the type of data we got. So this is just looking at nodule number for now. And at least for me, this was a bit of a surprise. So we had sites from the 3 stations that either had a high amount of residual Rhizobia or a low amount of residual Rhizobia present. And we sort of expected that when there was a lot of Rhizobia already there, maybe we wouldn't see the response to inoculant. But if there's not a whole lot, we should probably see a nice response to the inoculant. In this case, we're quantifying how many nodules those plants are putting on. And really, the take-home message here is that it's context dependent, depending on the crop. So like I mentioned, these are different pairs of organisms, and maybe we have to start looking at every single crop individually and seeing how they're responding. But in the case of peas and lentils, even when residual Rhizobia were low, we did not see an increase in nodulation coming from applying that inoculant. The black is a Hettinger with low residual Rhizobia. We almost had no nodules there. That's just a great example of why high residual nitrogen is a problem. That field had a little bit too much nitrogen left in it and nothing but nodules on it. Chickpea was a different story, and we did see increased nodulation in response to especially the granular product that we applied in the chickpea fields. Now, of course, the really important thing is, do we actually see yield responses to inoculation? And really, this is just showing the data from Williston, although we just got the data in from Carrington, and it seems to have a similar result, where we saw these yield benefits in chickpeas when we inoculated, even if residual Rhizobia were present, but we weren't really seeing much in the ways of yield response in peas or lentils. And so this is really, an issue when we try to think about all our legume crops and wanting to make sure that we're maximizing symbiotic nitrogen fixation there. Because of an old concept in the literature called the rhizobium competition problem. So this problem states that the natural rhizobia that exist in fields are less adapted as really efficient nitrogen fixers. And when they form the nodules rather than the inoculant, we don't get the benefits that you would expect to get from that high levels of nitrogen fixation from a particular inoculant rhizobia that's been selected for that elite performance. So that is to say, there is a difference between an effective rhizobia and an ineffective rhizobia. And just because you have a nodule on that crop does not mean it's doing the best it could be in terms of delivering nitrogen. And when these ineffective rhizobia are there in our soils and they're locally adapted to really thrive there, they can outcompete those inoculant strains. So this has been a concern for a long time. And I think we have a nice opportunity to sort of dive in deeper to see whether we think that's happening. Now, we don't know yet in the case of these field trials, whether those rhizobia that were there, are they beneficial or are they not? Are they effective rhizobia or are they not? And understanding that would help us to think about how to maximize this symbiosis in agriculture. So if they are beneficial, well, that's great news from a farmer's perspective. That means we can use our precision ag tools to simply guide inoculant decisions. And as long as you've got a good population there, you don't need to inoculate. And maybe we can even find ways you can nourish those rhizobia and maintain healthy populations that can keep your nodulation nice and high without applying inoculant. So that would be the good case scenario. The bad case scenario is if those local rhizobia are not beneficial. In that case, when they form the nodules, we're not able to get the benefit from symbiosis that we would like in order to get good yields and incorporate nitrogen into our fields for downstream crops. So if that's the case, then the question shifts and it starts to become a technology question. How can we start to make sure that the inoculants we apply are really competitive and they can outcompete these natural strains and actually get into the root nodules? So to get at that question, what is the quality of the rhizobia that just exists there naturally? We did a big project that was funded by the Northern Pulse Growers Association, sampling pea rhizobia from across the state. I had 3 amazing undergrads, Mia, Grace, and Natalie, who actually developed a collection of 1,000 rhizobia over one summer from sites all the way across North Dakota, which has this great transect of all sorts of different soil types. And using this big set now, we're trying to ask that question of what is actually the quality of these rhizobia that are just there existing in our fields. And this is the type of data that we get. So in this case, we've done an experiment called a shoot dry weight assay. We've grown the rhizobia one by one with a pea in media that lacks nitrogen to try to really emphasize or make them dependent on the rhizobia for all the nitrogen they get. And the result is that the bigger the pea gets, the better quality that rhizobium that it has there with it is. And what we see is basically a continuum. So there's no easy answers. So some of the rhizobia were highly beneficial, whereas others were literally not beneficial whatsoever. They were no different from applying water. So they look like this tiny pale green plant that's shown in the picture here. And this is kind of representing the continuum that we see of the quality of these strains. So really a complex situation going on then in the soils in terms of how good the rhizobia are. So then the big question for us was not just all the strains we could isolate, what was the quality, but which ones are the most competitive? Which ones are the ones that tend to form the root nodules if you actually apply, or if you're actually planting there and they're all existing there in the soil? To assess that, we've used a really amazing technology we developed during my time at Oxford, where we can actually mark the rhizobia with these molecular barcodes. That lets us use similar technology to what we would use to sequence the microbiome, to sequence all of the rhizobia that exist in the root nodules of a plant at a time. At the same time, it expresses this protein called green fluorescent protein, which actually makes the nodules glow, and it makes them glow brighter according to the quality of the rhizobia in the nodule. This is kind of a game changer for us because historically, looking at competitiveness of rhizobia, which ones actually are better at forming nodules, was really slow, and you have to do it one by one, and it's really painstaking. Here we can apply hundreds of rhizobia at a time and get a sense of which ones of those hundreds are actually the most competitive. So this is just showing a P that we would have pulled out of one of our experiments. This concept that each nodule kind of has a molecular barcode in it, so we can see which Rhizobium strain made it into the nodule. And this is just kind of showing this cool glowing nodule that happens. And the brighter those nodules glow, the better the quality of the Rhizobium in each nodule. So this is really trying to create throughput to try to ask big questions about these large collections of Rhizobia. This is an example of some of the data we got out of it. So we have soils on the y-axis. These soils represent different soil types going across the state. So very, very different pHs, different organic matters, different textures. And on the x-axis is a subset of 100 of those different strains. And the brightness of the heat map here is basically how competitive each strain was in the soil. I was really surprised by this data. I actually expected that different strains would be more or less competitive depending on the soil that we looked at. And we did see that a little bit, but what we also saw is there was clearly some really elite strains in terms of competitiveness, in terms of their ability to beat out all the others to actually form the root nodules. And you can see that here. We've got 4 or 5 there that are universally quite clearly better than the others. So when we start to map those back onto the quality of the strains, what we do see is an enrichment of the Rhizobia that are poor quality tend to be more competitive. So that's a big concern, right? So this is the bad case scenario where the natural Rhizobia in the soils actually aren't of a very good quality, and they're not helping to promote the plant growth and to give it the nitrogen that it needs. Now, there are some that are good, and so you can see that purple arrow, maybe the orange arrow on the other side of the graph. And this is kind of hopeful and encouraging, and this may actually be, an example of from our program where we might take that strain and look at could we actually make an inoculant out of that and get a better response to inoculation when we go back into the field, because we have this highly competitive, highly effective strain. All right, so that kind of summarizes the data I wanted to show you about our work in Rhizobia. Just in summary of that, we're wondering, should farmers inoculate, trying to get a deeper understanding of that using precision ag tools. And we're looking towards how can we overcome this Rhizobium competition problem where the natural Rhizobia may not be high quality, but they're very, 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 very competitive for forming root nodules. Okay, I'm going to switch gears now and talk about nitrogen fixation in cereals, just checking the time. So I just want to cover briefly the strategies to try to get nitrogen to cereal crops, which can't carry out this symbiotic relationship, and I'll briefly discuss emerging biologicals that are becoming on the market as tools for farmers. So I worked in this area in the past in a postdoc. This is really blue sky, moonshot stuff, where if we could get a cereal that can fix its own nitrogen, that would be transformative to agriculture. And we do still do some of this work in my lab. There's different approaches different groups are taking. Everything as wild as can we actually express the enzyme for nitrogen fixation in the mitochondria or the chloroplast of the plant itself, so it doesn't even need a microbe. To what I view as a more practical solution, can we actually just get the cereals to make root nodules for Rhizobia? Although still a lot of work's got to be done before we're going to get there. And probably the nearest term, and this bridges into the biological products, is can we actually just take members of the cereal microbiome, not necessarily Rhizobia that make root nodules, but just bacteria that thrive on or in the roots or the shoots of plants, get them to fix nitrogen and give a bit of that up to the cereal. Now, this is never going to do what Rhizobia can do in terms of providing all the nitrogen a plant needs, but it would be a boon for sure and might reduce dependencies on fertilizers. So these emerging biologicals and some of the research we did as well looks at these what we call associative diazotrophs. So these are bacteria that can fix nitrogen, and they do not need to be in a root nodule to do so in the free living state. Now, they can be associated with the plant, but it's important to remember these are going to be members of a very complex microbiome. Rhizobia get a dedicated environment in the root nodule where they can live in millions, in huge numbers, right? But in this case, these diazotrophs have to compete with millions of different microbes that are also living in the microbiome of the shoot and the root of the plant. It's also questionable whether they're truly symbiotic. Are they actually in it for benefiting the plant? Generally, they're probably fixing the nitrogen mostly for themselves. Nevertheless, the question is, can we get some benefit from these free-living diazotrophs? Even though we know they'll never completely replace fertilizers, maybe there's some value to be gained from exploring this approach. So these emerging biologicals, I want to emphasize that they're not a replacement for fertilizer. They cannot fix nearly enough nitrogen to replace the requirement for fertilization. And actually, I think the most promising suggestion of how to use them is really as a supplement for fertilization, to look at having this biology on your plant that as the fertilizers are running out that you might have applied in the spring, they can pick up and fix a little bit of nitrogen as the plant starts to become nitrogen deprived in the middle of the growing season. So they have this potential to fill the nitrogen gap when the plant starts to have used everything that was there during the middle of the growing season and boost yields that way. So one of the products that I believe is available now in Canada is Invita from Exotic Technologies. This is probably the one that's been in development the longest. I remember seeing this company many years ago started working on this strain. This is a bacteria, Gluconoacidobacter diatrophicus, that was isolated from sugarcane in Brazil. It's suggested to systemically colonize both the roots and the shoots of the plant, and hence it can also be applied either in furrow or as a foliar spray. It's advertised to work for many different crops, not even just cereals, and that the bushels per acre they're suggesting for corn is about 5.7 bushels per acre benefit that you would get by incorporating this into your nitrogen program. Utrecia is another one from Corteva. This is Methylobacterium symbioticum, very different bacteria. This is a specifically phylosphere or leaf colonizer. Hence, its application is only foliar. Again, it's advertised to work for many, many crops. Yield benefits still in that same range of 5 bushels per acre for corn. But you can see it's a similar concept. This microbe ideally is colonizing your plant. It's fixing some nitrogen, and especially when nitrogen runs out in the soil in the middle of the growing season, maybe you get this yield bump. Another one I wanted to cover is one that I don't believe is currently available in Canada. It's a US product, but this has really been taking off here, proven by Pivot Bio. So this is actually a mix of 2 strains that were isolated from corn roots. It colonizes the roots, but what's really kind of wild about this one is it's actually been gene edited or genomically remodeled, as they put it. So they've actually made targeted deletions and mutations to these strains to try to reduce any natural repression of the nitrogen fixation and get them to fix as much nitrogen as possible. So this might be a promising way to sort of overcome this problem that these bacteria aren't necessarily in it for the plant. Well, maybe we can kind of force them to be. And again, they kind of advertise around a 5.8 bushels per acre increase from this product. So when we're thinking about these products, I just want to emphasize, the biggest thing about these products is that both any university field trials I've seen and even the data that you see that they put out about their performance shows that they're not necessarily something you can rely on. So they're variable in their performance. Sometimes they work great, sometimes they don't. And you see that where you see, these curves that show very different levels of performance depending on the field that they were applied in. Probably the reason for that and the challenge here is that they have to colonize and persist across all these different crops, different environments, and they're not necessarily adapted like the local microbiome would be to thrive in the plants. Again, getting them to give up their nitrogen is also a challenge, and this may be able to be accomplished by engineering, like in the Pivot Bio strain. And the other thing that I really want to make sure farmers are thinking about is that these are not fertilizers, right? These are living organisms. So shelf life, thinking about that is really important, and thinking about applying it in a way that's not going to be harmful to the organism is really important. So things as simple as the water that you use, making sure that's not too alkaline or not too acidic is going to be a really important consideration if you're thinking about incorporating this into your program. So just in summary, are these an opportunity to maximize yield? I think, yeah, they are an opportunity, but they're being a new technology, there's a lot of work to be done to vet these new technologies. And this is really something I hope a lot of agronomists in the audience or in the room are thinking about. I think there's opportunity too for farmers to just do some testing themselves. And, if you're going to use these for the first time, always, talk to an agronomist about how to incorporate an untreated check, or even talk to the company. Sometimes they'll actually help you with that. So you can see whether you're seeing these benefits on your farm, because really there is that variability, and it's important not to just kind of blindly go in and apply them and hope they're working. You want to be able to see that evidence that they're doing something for you. And I think as a community, as we start to understand these better, we'll get a better sense of whether these are really, an important tool for agriculture going forward. That's it. I just want to acknowledge the collaborators at Research Extension Centers, Audrey Khalil, Mike Osley, and John Rickardson. And really my whole lab at NDSU, a lot of the work I showed today was done by Lucas Andes, and the 3 undergraduates I mentioned. So thanks a lot. Hopefully I got time for a question or two before lunch, but thank you again for the invitation.
Curious about the energy biochemistry of the plant, and in particular, doing quite a bit of work on N2O emissions. And we look at soil nitrogen transformations, and it seems like a shame to me where we take all of our fertilizer in and it is oxidized to nitrate, taken up by the plant, and the plant has to then reduce all of that nitrate back to feed into its protein synthesis machinery. So my question is, in nitrogen fixing plants, how much of the benefit is because the plants no longer have to spend that energy on nitrate reduction? Or am I way off base here?
Yeah, I think that's an interesting concept, thinking about the different forms of nitrogen and what's more energetically intensive for the plants to handle. I mean, so in nitrogen fixation, these are made as it gets converted to ammonia, right? So it's ammonia that the plants are getting from the nitrogen fixation process. They turn that pretty quickly into amino acids or into these things called ureides that transport the nitrogen and then get converted into amino acids. Probably it is a little bit more efficient. The thing you got to remember, though, is that the plant also has to spend a bunch of energy on fixation. So I don't know that it's more efficient than converting nitrates, because they have to fuel the process and feed the rhizobia the ATPs that they use to fix the nitrogen. So I think actually there's maybe more likely to be a slight cost in terms of the nourishing of the symbiosis than necessarily a net gain.
Very good. Thank you. We do have several online questions, so we'll go over to Tammy for one of those.
Oh, if I have to choose one, then I'm going to, we'll start with a really easy. What does a quantitative PCR assay Rhizobia test cost?
Yeah, so we haven't rolled it out for farmers. We're just doing it for free right now, but I'm guessing it's going to be costing like 10 or 20 bucks, something like that. So a lot less than the, 1,000 you might spend on inoculant.
Well, 120 bucks, that's only 6 bushels of soybeans. So
10 or 20, not 120.
next
Yeah,
question.
10 or 20. Okay, so then there was a question about IDC and soybeans. And if IDC negatively affects the symbiotic relationship in soybeans, would higher rates of inoculant somehow help to combat that IDC?
Yeah, that's a good question. Maybe. Maybe. I'm not sure yet. I think this is something that's not really that explored. It's stuff that's come up through our work on IDC and actually looking at microbes that might be able to help with IDC and looking towards, could we actually get to an inoculant of a rhizobium with a microbe that would help reduce IDC at the same time? Higher rates, maybe. I would think that the more beneficial thing would be to put a bit of pressure on industry or on us to try to find strains that are less sensitive to the IDC. So the ones that the main one for soybeans right now actually doesn't produce any of these molecules called siderophores, which are used to acquire iron when it's low. So I would assume there's some natural diversity that might be exploited to be a bit better in IDC conditions, but yeah, it's largely unknown at the moment, I think.
We're going to go to another question in-house here.
Hey, my question's about sort of these expensive inoculants versus the cheaper inoculants. Is it worth the money to pay the money for these more expensive inoculants? Like, are these strains of Rhizobium better than kind of the cheaper inoculants?
Yeah, I mean, I am currently not a big believer in the inoculants that have other microbes added into them. So in my experience, I haven't seen much difference if you just have a regular Rhizobium versus a Rhizobium plus azospirillum, although this is something that in Brazil has shown a lot of benefit. The biggest cost difference for me, I think, is the type of inoculant, right? So liquids tend to be a lot cheaper versus granular. And there you got to think about basically your ability to take care of the inoculant. So granulars are good if you need to be flexible and you need to have that sitting in your shop until you go to apply the inoculant, because they have a better shelf stability, but they are way more expensive. If you know that you can go and grab that inoculant, put it on your seeds and get that in the ground that day, then I think liquids will work just as well. And they tend to be, $3 an acre versus $10 an acre. So yeah, that's kind of what I would think about that.
We're going to, Tammy, do you have another question online?
I do. And Barney, there's a few more. If you are willing to answer, then we'll circulate out those answers. But let's talk about third-party research with some of these new things like Invita, Utrisha, and Improven. Are you doing it? Is there someone doing it? Where is it at? And how do we start to compare those potential benefits to the cost per acre?
Yeah, I think third-party research is super important there. My lab and the structure of my group is not really set up to do this. I collaborate with agronomists to do all of our field trials. But I do know that there are agronomists at NDSU that are starting to test these products. Dave Franzen, I think, has been testing them, as well as a few of the scientists at the RECs. So hopefully some information about how well they're working will start to come out. I think, again, the challenge we're facing is really that variability. It seems like you see a trial and like nothing happened, and then you'll see a trial and they saw some benefit, and we just need a lot more research done. And I hope that agronomists at the different institutions that have that ability to go out and test products in the field will definitely take up that challenge to try to get a better sense of whether these things are beneficial.
Okay, I think we do have the yield data on that slide from Lenore, Manitoba that you showed there. So that may get people to a poster.
Yeah,
One
there
more
you go.
question.
One more question, or maybe more. Is there any legacy to some of these products like Evita or Proven or these non-leguminous inoculants, so that we can maybe not count on the first year with a response, but maybe on the fourth year if it's still lingering in the soil?
Yeah, that's a good question. My opinion is that the answer is no. I think we're already doing pretty well to take something that's been isolated from sugarcane in Brazil and put that on corn in Manitoba, and even expect that to stick around for the growing season, let alone for multiple years, getting through the winter and all of this, where it's got to compete with all the rest of our natural soil microbes that are much better at already persisting there. So I don't have data on that. It's something I am kind of interested in looking at, but my gut would be, I would definitely not count on them sticking around.
Okay, with that, Barney, the questions will continue. I think we can forward some to you online. I expect the Manitoba Pulse and Soybean growers, when you get your green card, are going to get you on a program at some point. But in the meantime, let's thank our final speaker here.
Thanks a lot. My pleasure.